our bone has the inherent capacity to regenerate itself throughout the adult life. However, in case of large defects, when the defect size goes beyond a critical limit, the bone is not able to heal itself. Rather, it needs a secondary therapeutic intervention. For a long period of time, these defects were treated with the help of transplants like isografts, autografts, and xenografts, but these come with the limitations of their own. Therefore, advanced strategies for the fracture healing has been come into play, which involve the use of bone tissue scaffolds, biological strategies by using cells, and physico-mechanical strategies. So our work is mainly focused on the combination of these three strategies. Well, we all know the importance of stem cells in our body, and in case of bone defect healing, stem cell-based therapy has been used from decades to treat the bone fracture. However, the direct use of stem cells at the target site is involved with a lot of risks. That is why another strategy has come into play, where instead of using the cells, the cell-derived secretory factors are used. So that is why it is known as cell-free strategy, and exosomes are striking examples of that. So coming to exosomes, these are tiny vesicles measuring about 50 to 200 nanometer in diameter. They are secreted by most of the eukaryotic cells by following an endosomal pathway. Because of their lipid bilayer structure, they can be easily uptaken by the host cells, thereby regulating a lot of functions. If we look inside the exosomes, we can see a lot of important information are encoded in the form of microRNAs, mRNAs, proteins, and lipids. That is why, to achieve our hypothesis, we isolated exosomes from bone cells. We took the cell culture supernatives and we isolated exosomes with the help of ultracentrifugation. Then, to confirm if the isolated particles were actually exosomes or not, we characterized their size, surface charge. We saw their surface morphology with the help of electron microscopy. Then we checked the presence of the exosomal marker proteins with Western blotting and flow cytometry. Interestingly, we saw that when we treated fluorescently tagged exosomes to the host cells, the cells were able to readily uptake the exosomes with increased time of incubation, which showed their remarkable cell uptake ability. So now we wanted to see what is the effect of these isolated exosomes on osteogenic mineralization. For this, we took different concentration of exosomes and treated them with the recipient bone cells, and we saw that these exosomes enhance cell proliferation and cell migration in a concentration dependent manner. Furthermore, we saw that the osteogenic marker proteins such as ALP, collagen, RUNX2, and also the genes were significantly highly upregulated in exosome-treated cells. We also screened for the ALP activity and alizarin calcium deposition with alizarin red staining, and the data suggested similar kind of results as that of our previous analysis. Well, now, the naturally occurring exosomes are not suitable. Despite of these advantages, they are not suitable for therapeutic application. Why? Because bone injury is a localized injury and exosomes are just vesicles. So we thought, why not integrate these exosomes on a scaffold matrix which will hold these exosomes, give it a proper shape, and regulate their release at the target site, thereby giving a localized treatment. That is why we synthesized polymeric scaffolds with the help of UV cross-linking by taking chitosan and collagen. To confirm the level of cross-linking, we performed proton NMR. Then we also saw from scanning electron microscopy that exosomes as well as the cells were able to adhere to the surface of the scaffold. And the exosomes were able to release with time, and the scaffolds were able to self-degrade. To confirm, to see the applicability of our scaffold exosome construct, we created a rat model of critical size defect of 9 mm at the cranial region and implanted our scaffold construct at the defect site. And after 8 weeks of implantation, we collected the skull and performed X-ray images and micro-CT scanning. From the images and the graphs, you can clearly see that significantly higher amount of new bone growth was there in case of our treatment group as compared to the control. And in fact, it was 3 times higher than that of the control. To further validate our data, we decalcified the connected scaffolds and performed histological sectioning followed by staining with He Mason's trichome and IC. And these data also supported our previous findings from the micro-CT, which suggested that mature bone formation were there in case of the scaffold exosome and scaffold cell treated. Also, I would like to add one point here that you can see that in case of scaffold exosome treated group and scaffold cell treated group, similar level of bone regeneration we found. And this signifies that exosome itself is competent enough to create bone regeneration, to have new bone formation, as like we don't need any cell to be present there. So this proved our cell-free therapy. So to summarize, we found that bone cell-derived exosomes, when integrated with proper matrix, have therapeutic potential in bone regeneration, and they could have more potential in upcoming tissue regeneration field. I'm glad to share that we have filed a patent with this work, an Indian patent recently. So finally, I would like to acknowledge my PI, Dr. Bhavani Das, ILS for infrastructure, GIFT for registration, CSR for my fellowship, my entire lab group. I would like to give my heartfelt thanks to the organizers of this international conference, Utkal University, Department of Zoology and Zoological Society of Odisha for giving me this opportunity. Thank you all for your patient hearing.
Thank you very much for a great presentation and finishing within time. You still have almost 2 minutes, so you deserve time for one question. If anybody has a question. Yes, ma'am.
Yeah. No, ma'am. That is what. Yeah. Yeah. That is a very important question that everybody asks. That in case of since they are coming from a biological origin, the homogeneity is not same all the time. But what we are doing, we are taking already enough amount of exosomes as a starting material. So it is not like every time we will not get that much of information from the exosomes. But definitely there will be enough quantity of that information present inside the exosomes to have that that activity. Ma'am, right now we are taking exosomes from cell culture media. We are culturing mouse periosteoblast cells and taking exosomes from the media. Supernatant.
So we have to leave there. Thank you, Susmita, for great presentation again. Thank you. This one. So let's go to next presentation, Sandeep Kumar Mohapatra, please. Is Sandeep here? Mohapatra. Okay, let's try Narayan Panda, next presenter. He's presented the board. Okay. So is our next presenter, Sandeep Mohapatra. Please come on the stage. Sorry about the confusion created by the chair.
Good afternoon, everyone. I am Sandeep Mohapatra, a PhD scholar from Department of Zoology, Berkeley University. Topic is diversity of parasitic isotope family cyberspace on the position. So, animals interact in various ways, which is inclusively known as symbiosis, and it is broadly classified into positive interaction and negative interaction. The positive interaction includes commensalism and mutualism, where at least one species get benefited, and the negative interaction includes amensalism, predation, and parasitism, where at least one the species get negatively affected. The isopods are crustaceans coming under the class Malacostrata, and they are found in marine environment, freshwater environment, and terrestrial environments. And they are the only crustaceans which can be found in terrestrial environments. The marine isopods are again classified into free-living isopods, parasitic isopods, and micro predators. The free-living isopods feeds on detritus. The micro predators are actually like mosquitoes. They attach to fish and other animals to get blood, and when their belly is full, they detach. Parasitic isopods can be found attached to its host for the rest of life. Then why should we focus on thymoid isopods? Because they are responsible for problems like gill filament and gill vacuole get damage, inflammation, irritation attached to the surface, which ultimately leads to respiratory problems of fishes. They also cause death in fish fry, weight loss, which ultimately leads to economic loss to the fishermen. And also to know more about the parasitic behavior and adaptation of thymoid isopods, and they are so mysterious that we do not know much about how much they cause the infection to the host fish. Worldwide, most of the species are described from the Western Pacific region, and to polar regions, the number is greatly reduced. From India, we have 48 species of thymoid isopods, and from Odisha, we have 2 species which attach to the buccal cavity of isopods, 4 species which attach to the branchial cavity, and 8 species which attach to body surface of host fish. We visit the different fish landing centers and check the host fish on the body surface and the gill chamber, also the buccal cavity, and using a pair of forceps, we remove the isopods. There are 2 species of buccal attaching species. One is Lobothorax which only affect one host fish, which is Leptoracanthus avala, or the ribbon fish. And the Thymophila indica is actually attached to various host fish, and it is much prevalent in Chilka Lake. The branchial cavity species of Mosasaur grenadi, Mosasaur quality is only found in host fish, Strombylura lura. Norilica indica, here you can see it's attached to a host fish, which was Rastrolator canduta, and it is also economically very important fish. Jorema hawaya and Agamammalai is found attached to the gill chambers of Clupidae fishes actually. Then there are body surface attaching species, Narosilla sundaica, Narosilla sigani, Narosilla aris, Nerothila depressa, Nerothila phyoclura, Nerothila orbigni, and Nerothila burove. And there is a genus of Anilocra immediata. All these exoparasites are actually interesting because they attach to different regions of host body. Nerothila sundaica is mostly found attached to ventral region of the host fish. Nerothila sigani and avis is found attached to the caudal pedicle of the fish. Nerothila depressa and phyoclura overlapping region of attachment, which attaches to the mid portion of the body. Nerothila orbigni is found attached to the fin of the host fish, which can be the dorsal fin, the pectoral fin, and also the caudal fin. Nerothila burove attached to the dorsal surface of the host fish. Anilocra dimidiata is found mostly attached to a region near the eye. Then what are the problems which are associated with identification of isopods? There is an intermediate stage during the development of the parasitic isopod. After the completion of the juvenile stage, the male stage is there, and in between from male to female, there is an intermediate stage. The first half of the individual is male, and the last half of the individual is female. And much of the descriptions of the species is based on the female character actually. So you cannot identify this individual using the female characters because the first half is male. So it creates a problem. And there is sexual dimorphism. The male is very smaller and the female is very larger. So they sometimes get confused that it is a different species. Then there are some confusing species like Nerothila aris and Tigani, which have much of the characters having overlapping, and also the DNA sequence is also not available in NCBI, so we cannot differentiate them. Then a main problem is language barrier. Much of the descriptions are in French, and we cannot actually translate it using Google Translator. And if it gets translated, it is actually not much informative. Then there is region-wise morpho differences. In different ecoregions, the isopods have a little different body character. So sometimes they interpret it as a different species. And these are some results we get. We reported some new state fauna isopods and also some new host records of these parasitic isopods. And this Nerothila orbigni was reported first time from India, and it's from the Chilka Lake. And it's Nerothila kisra, it's also a new record to India. And apart from the taxonomy, we also found that the male, female, and the juveniles actually have different spatial needs. And the females mostly lie on the vocal cavity, males behind the female, and the juvenile is mostly found on the gill cavity. It reduces the competition in between the same species actually. I'd like to thank the director, Jitniti Banerjee, and TIASRST for providing necessary funding and the lab facility. I'm also thankful to my guide, Dr. Jagdish Stage, and my co-guide, Dr. Vasudev Tripathi, and also our officer in charge, Dr. Anuma Pasra, for the support and guidance. And I would also like to thank my lab mates and also the local fishermen, which provide more information about the isopods. These are the references. Thank you.
Thank you very much. Again, you finished right in time. Wonderful. But we ran out of time, 10 minutes, so we will invite the last presentation before lunch. Thank you again. Give him a big hand. Narayan Panda is the next presenter. Please welcome Narayan. Thank you.
Good afternoon, everyone. I am Narayan from National Institute of Oceanography, CSIR. Our institution is specially focusing on the ocean study to explore, understand, and protect the ocean environment and the organisms. So, as per this study, my topic is distributions pattern of benthic megafauna in 4 atoll lagoons of Lakshadweep Island. So, benthic megafauna in the oceans, we can see that if you see ocean or sea, there is a 2 type of zone. Like one is pelagic zone, another is benthic zone. So, benthic is studying the bottom part, which is from the intertidal zone up to the deep sea. This is benthic environment. The organisms which are present in this bottom part of the ocean, they are called benthic organisms. So, based on the classifications, they are divided into different types, like based on habitat, based on nutrients, based on size. But in this study, we will focus only based on the size. There are macrobenthos, meobenthos, and microbenthos. So, we will focus only into the meofauna or meobenthos. That size ranges between 63 to 500 micrometers. In some literature, you can find that from 1 to 1000 micrometer, but based on the sieving unit, we are focusing only from 63 to 500 micrometers. So why we study these organisms? Because they have, as per the previous publications, they have different ecological importance and different role of meofauna in the ocean. So let's travel into the ocean island. That is, this is our study area, that Lakshadweep Island, which is comprised of 36 small different types of islands, but we are focusing only 4 islands, that is Agati, Kavarati, Bangaram, and Paramalpar, which is southwest in the Arabian Sea, which is present. So you people know that a healthy coral environment. These all islands are surrounding by atolls. That is coral is breaching barrier and atoll. 3 types of coral reefs are present, but these islands are surrounding by atoll reefs. So a healthy coral environment provides the highest habitat heterogeneity, and now the islands are subjected to development purposes like construction, storage purpose. So lots of construction works are going on. So every day, without any proper documentation, lots of organisms species we are losing. So because of the importance, we study these organisms. Our objective is the abundance and distribution of megafauna in 4 atoll lagoons of Lakshadweep. These are the 4 atoll lagoons. This is the methodology. We collected the samples by diving into the island and by using the coral. And after that, on board we did sieving. Then we preserved the samples under MgCl to 7% and neutralized the formalin. For MgCl2 for the anesthetic reagent. After that, we did the processing of the samples in the lab by serial sieving, 563 micrometer of sieving unit. Then we sorted the organisms under the stereoscope microscope. Then we preserved the what are the different taxons we got. These are the different taxa like nematodes, polychaetes, copepods, ostracods, nematodes, bivalves, and oligochaetes larvae, and other mollusca, nauplius larvae, and acarids, and other organisms. So coming to the result section, what we found in this result, overall compositions of the organisms and their abundance we studied. Like in overally, we can see that nematodes are showing the dominant group, followed by the crustaceans, then foraminiferans, then polychaetes. Based in this, they are dominating, especially nematodes are showing dominant group. But in comparing to islands, the Bangaram island showing the highest dominance, and followed by Agati, then Paramalpar and Kavarati. Why this type of results is showing? Because Kavarati, I am expecting that I am not just I did the 7 months of study. I am not expert in this field. Just initially studying the work. So I am expecting that Kavarati is the administrative island of administrative island. Lots of construction work and transportation and the anthropogenic activities is very high in this island. So maybe because of that, because here organisms are getting affected, and but the Bangaram is a tourist island. Now it is, but it initially from 2 years before it is tourist work started. But in comparing to islands, organisms within the island, we found a variation, that is the crustaceans group, which is found the dominant in Agadi and Paramalpur both islands. Where nematodes found the dominant in the Bangaram island, and another is that is the foraminifera shows the dominant in Kavarati island. So island to island abundance also we see the variation, and also within the islands also dominance also we see the variation. These are the hydrological parameters which predict during sampling by using the CPD, the salinity, temperature, and pH and dissolved oxygen. You can see that there is no such variation within these hydrological parameters. That is, salinity is 34 to 35, and temperature is 28 to 29 degrees Celsius, bottom temperature, and pH also, and also dissolved oxygen. Within the each island and between islands, there is no such variation. Thank you. So from this, it is a pioneer work. There is no such work from the islands before. Some papers are there from intertidal zones, but there is no study from lagoons. But this is the pioneer work. But this initially basic work also we found more than 10 number of different taxons of organisms. So and while analyzing the samples also we found there is numerous number of genus and species, genus and families also there. But I am not expert in this field, so initially I just started the work. I am unable to identify all the groups and species, but definitely I will continue this work. And but as per the results, the variation I am expecting from the publications, previous publications, it may be due to the sediment texture and or availability of the organic carbon content or food availability or other purposes maybe. But homogeneous distribution pattern in hydrological parameter we observed, because this is the open ocean in the middle of the ocean, and this is oligotrophic environment. Especially productivity is high, very less, but the dissolved oxygen and bottom oxygen content is very high. So nowadays the islands, as I said before, islands are subjecting to the development proposals. So this type of study is furthermore needed to identify and to better understand the ecological importance of these organisms with relation to the corals and so on. Thank you.
Thank you very much for your presentation, again finishing in time. So he deserves, because he saved his time, so if you have any burning questions, please go ahead and fire up. Any questions?
Study like pH and temperature and salinity, the presence of humans on the islands, has it ever affected this?
Yes, ma'am.
Fauna or your study subject?
Yes, ma'am. Actually, this all the 4 islands based on human habitat islands, but only one that PMP, that is Paramalpur island. This PMP island, which is a submerged island. There is in rainy seasons the water will be the over, and sometimes the water will be in summer seasons. You can find the water, this island. So this is not human inhabited island. So there we didn't found any such type of effect. But in Kavarati Islands, that it is a high population island. But here we found that organism number is very less. So I am expecting that maybe due to the anthropogenic activities or oil spilling seeds, number of seeds is transportation is very high. That I am expecting that, but I am not sure. Yes. Sieving unit. Yes. Yes, ma'am. Our lab is togetherly we are working benthic and microplastic. Both we are working, ma'am. But no, ma'am. We observed that another part of work, so I didn't add it here. That was also. I am also focusing that microplastic inside the nematodes. Especially, we are focusing to that. Thank you.
Very, very good. We have to leave there. Thank you again. Give him a big.
Thank you.
So now may I request all the three speakers to come up on the dais so that I would like to request our chairperson, Professor Parati, to present the certificate of participation to all the participants. Please.
We have is Pandit Mama. Next is Narayan.